Thank you for that introduction. And yeah, thank you everyone who is here today, especially those of you who are up very early and not on the East Coast. I appreciate it. So I'm going to be talking about my thesis research that I defended last month that we're also currently working on finishing up in the Lombardis lab here at Columbia. And it's focused on better understanding the molecular basis of solid phase separation in olfactory transcriptional hubs. And before I get into that, I just want to take a brief moment to overview what we study in the Lombardis lab. Now, very broadly speaking, we all study various facets of our sense of smell or olfaction. Now, olfaction is mediated by olfactory receptor proteins, which I'll be abbreviating ORs. Now, ORs function by physically binding to chemical odorants from the external environment. Upon binding to these external odorants, OR proteins undergo a conformational change and start a signaling cascade that allows the neuron they reside in to signal to the nervous system and ultimately the brain. But what I'm really focused on studying and talking about today is better understanding how OR transcription is regulated at the molecular level. Now, we're really interested in this because of the idea that olfactory receptor genes are transcribed in a monogenic monoallelic fashion. Now, this means that every olfactory sensory neuron must express a single allele of a single olfactory receptor gene. However, this really requires a huge amount of regulation because mice have over 1,000 OR genes scattered across the chromosomes in various clusters shown here in red. So this singular transcription means that an OSN must express that single OR gene, but it also has to simultaneously prevent the transcription of hundreds of other genes scattered across the chromosomes. And I think if we better want to understand how this singular expression is achieved, we need to look at where the active gene is transcribed. Now, we know that the active OR gene is transcribed in the OR transcriptional hub. Now, the transcriptional hub has several components. It contains the Greek island enhancers, shown here in yellow. It also contains the transcription factors LHX2 and EBF1, along with their cofactor and architectural protein LDB1. Now, what's really interesting about this transcriptional hub is how the DNA in it makes contacts with itself. Now, in the transcriptional hub, we know that several Greek island enhancers must converge together and form very precise contacts with each other. However, these contacts span extremely long range, up to 80 megabases, and they can also form contacts with Greek islands on other chromosomes in trans. So the OR hub contains Greek islands forming very precise interactions in cis and in trans, and they must be maintained throughout the lifetime of the OSN for stable transcription. Now, what's also interesting about this system is that the Greek island intergenic enhancers are enriched in binding sites for the transcription factors LHX2 and EBF1. However, the promoters of the OR genes are also enriched in the same binding sites for LHX2 and EBF1. But we know through chip sequencing that the transcription factors bind widely to the Greek island enhancers, but not the OR promoters. And this fact really started my project. We kind of started by wondering what accounts for this robust transcription factor binding over the enhancers and not the promoters. And furthermore, what about these interactions stabilizes the transcriptional hub? And we started this project with a finding by a previous postdoc in the Lombardis lab, which was really interesting. And what he found was that the Greek island enhancers are enriched in a motif, which I'll be calling the composite motif. Now, this composite motif contains an LHX2 binding site placed directly next to an EBF1 binding site. What's interesting is the composite motif is enriched in the Greek island enhancers and not in the OR promoters or co-bound sites genome-wide. And in data I don't have time to talk about today, we also know that in vivo, our chip sequencing data suggests that LHX2 and EBF1 co-bind cooperatively to this composite motif. And so with this finding, we started this project wondering if LHX2 and EBF1 bind cooperatively to this composite motif in vitro and whether or not they form a complex. So to address this question, I purified LHX2 and EBF1 recombinantly in bacteria. I also purified their cofactor LDB1 in a truncated form. Now I used these recombinant proteins and I used electromobility shift assays to quantify how they bound to different DNA motifs. Now, what we're looking at here is a gel shift assay with recombinant LHX2, looking at LHX2 motif found in the promoters and EBF2 motif also found in the promoters. Now, we see that recombinant LHX2 binds to its individual motif, unsurprisingly. And we also see that EBF1 does not bind to LHX2 motif, also unsurprising. However, when we mix these two transcription factors together, we also see that they do not form complexes over their individual DNAs. And we see the same effect when we look at EBF1 binding to its own motif. However, the story changes when we start to look at that composite motif found in the Greek island enhancers. Now we see that LHX2 binds to the composite motif individually, and we see that EBF1 also binds to the composite motif. But when we mix these two proteins together, we see that a shift occurs, indicating that these two proteins can co-bind to this composite motif. And this was actually our first ever evidence in vitro that these two proteins can co-bind to this very small motif found in the Greek islands. And with that knowledge, we next asked ourselves if these two transcription factors had very different affinities for the composite motif of Greek islands. Now, to study this, we used gel shift assays again with those recombinant proteins. We quantified the binding of LHX2 and EBF1 to 3 different composite motifs found in 3 different Greek island enhancers. Now, when we do this, we see that LHX2 binds much more strong to the composite motif compared to EBF1. We see that LHX2 binds between 2 and 20 times better, depending on the motif that we look at. So with this dramatically different affinity, we next asked ourselves if these two proteins bind tighter together than their individual components. Therefore, we're also asking if they're binding cooperatively. So to address this question, we again used gel shift assays, but we compared the binding of the individual proteins to the complex as a whole. Now, the binding of the complex is shown in blue, and the binding of the individual protein is shown in black. Now, we were very surprised by this result because you can see that these binding curves look quite similar to one another. And so this told us that LHX2 and EBF1 do not have dramatically higher affinity for that composite motif together compared to binding alone, nor do they bind cooperatively when we quantify that. And that was actually very surprising for us because we assumed that this was like a traditional transcription factor model where one protein binds to a composite site and increases the affinity of the other protein binding. And so this surprising result led us to wonder if LDB1, that architectural protein, facilitates cooperative binding, and if that is sort of the critical third factor. Now, to study this, we used purified LDB1 protein. This is full-length LDB1. And we were really, again, surprised by the result that we got. So when we mix EBF1 and composite motif, we see that it binds as we're used to. And when we add in that LDB1, there is no binding, which is not surprising. But when we look at LHX2 binding to the composite motif, and then we add in LDB1, we see a complete loss of our LHX2 binding signal. Now, furthermore, when we mix LHX2 and EBF1, and then we add in LDB1, we see again a complete loss of all of our LHX2 containing complexes. And this is really surprising because it was showing us that adding in a full-length LDB1 is promoting the formation of higher order aggregates, but very specifically, only the aggregates are forming in conditions with LHX2. Now, for a sanity check, though, we purified the domain of LDB1, which we know binds to LHX2. Now, when we do a gel shift with that, we do see that it binds to LHX2 as predicted. When we have LHX2 and then add in that lid domain, we see a shift indicating they interact. We see that EBF1 does not interact with the lid domain, which we expected. And when we add in LHX, EBF1, and this lid domain, we see that the complex of all proteins shifts upward along with LHX2 alone. So this tells us that LHX2 does bind to LDB1 with the predicted domain, but something about full-length LDB1 protein is very specifically promoting the formation of higher molecular weight aggregates, which are preventing migration into our gels. And we were actually quite intrigued with this idea and decided to run with it and study how it might relate to the stability of the OR hub as a whole. And so with that, we started to look into phase separation as a possible explanation for the OR hub stability. Now, I'm not going to give a very long background on phase separation. I think everyone here has probably read a lot about it, as it has really been increasingly studied in many different systems. But in brief, phase separation occurs when molecules concentrate over a specific area and demix from their surrounding environment because that's more energetically favorable. Now, in vitro, you can quantify phase separation quite easily. I think this is a really beautiful example of two proteins in vitro which are forming liquid phase separated condensates. So with that background in mind and with our in vitro evidence, we decided to purify full-length fluorescently tagged proteins of LHX2, EBF1, and EBF1. We then mix these proteins together in physiological buffer in various combinations. We then transfer them to a standard microscope slide and image with fluorescence microscopy and DIC microscopy to visualize if no phase separation occurs, if liquid phase separation occurs, or something else entirely. So to start, we just mixed all three of our proteins together in solution with two different types of DNA. The first is what I'll be calling 5X enhancers. That is simply a linear stretch of 5 Greek island enhancers that were stitched together. We also compared that to a random plasmid DNA sequence of comparable size that was also linearized. And then, of course, we compared that to no DNA whatsoever. Now, when we mix all proteins together with no DNA, we see that they form very small puncta in solution, but it's nothing very compelling, right? However, on the other end of the spectrum, when we mix all three proteins together with enhancer DNA, we see a very dramatic shift in both the size and the morphology of the condensates. Now we see that at the same concentration, these same proteins start to form these very large, irregularly shaped, rigid structures. And they're significantly larger than when we add plasmid DNA, which does have a slight crowding-induced effect, but is not nearly as large and not the same in morphology. And you can see that reflected in our quantifications. So we were intrigued by the idea that enhancer DNA was driving these proteins to form very large, irregular condensates. So we next wanted to break that down and figure out what the components were doing individually. Now, this top panel that we're looking at is that same reaction from the previous slide. It is all three of our proteins fluorescently labeled, along with fluorescently labeled Greek island enhancer DNA. Now, again, we see that we have these very solid-like large condensates, and we see that they contain all three proteins co-localizing together. Now, we can also see that the end-labeled Greek island enhancer DNA, which is typically just diffuse in solution, is also fluorescently localizing to these structures, which led us to think that we're kind of forming an OR hub in vitro in a way. And again, when we remove that OR Greek island DNA, we see a complete loss of these structures. We see that LHX2 and EBF1 form puncta in solution which co-localize, and we see that EBF1 also forms puncta that do not co-localize. Now, furthermore, when we remove LDB1, when we remove that architectural protein, we see that we still have these very large solid-like condensates, but they are significantly smaller. And we can see that, again, LHX2 and EBF1 co-localize with that Greek island DNA. And LHX2 and EBF1 alone form these very small puncta. So in summary, when we add Greek island DNA to the transcription factors, we see the formation of very large solid-like condensates that become significantly larger in the presence of that cofactor, LDB1. Now, with this finding, we wanted to further probe what about that Greek island DNA is driving this very odd effect that we see. So to study this, we mixed all three proteins together and we compared their morphology with different DNAs. So this first panel shows all three proteins with a single Greek island enhancer called lipsy. And we compared that to a 1,000 base pair sequence upstream of Olfer-17, which contains its promoter. Now, when we look at this enhancer versus promoter DNA, we again were intrigued to see that only the enhancer was driving the formation of these large solid-like condensates. In fact, I want to remind all of you that the promoter DNA still contains binding sites for LHX2 and EBF1. The only difference being that the Lipsy enhancer contains that composite motif. And we were really intrigued by this finding, and so we wanted to really dive further into whether or not the composite motif of the enhancers is driving this effect, and what about the composite motif does this. To address this question, we made 2 mutations to that Lipsy enhancer that I just showed you. The first I'll be calling motif mutation. Now, this mutation will include a 5 base pair insertion between the LHX2 and the EBF1 half sites. So we're not removing the binding sites, we're simply spacing them apart. Now, that second mutation that we made was the motif deletion. Now, this is exactly what it sounds like. We deleted all of the composite motifs from that Lipsy enhancer. Now, when we compare unmutated Lipsy enhancer to those 2 mutated Lipsy enhancers, we see really different results. Now, Lipsy enhancer alone forms these large solid-like condensates, as we've been looking at. However, what's really exciting is when we add 5 base pairs in between the half sites of all of the composite motifs, we completely lose the formation of these large solid-like structures. Furthermore, when we delete the composite motif entirely, we also lose the formation of these large condensates, and they return to a state that's very comparable to when we have no DNA whatsoever. And we can see here in the quantification that the condensate is significantly larger with unmutated Lipsy compared to the motif mutation and motif deletion. Now, we found this really exciting for 2 reasons. The first is this result is telling us that the composite motif is what drives the formation of these solid-like condensates, but it also tells us a lot about this motif mutation that we made. Now, I want to tell all of you that this motif mutation does not reduce the binding affinity that the transcription factors have. When we perform a gel shift assay with this 5 base pair insertion mutant, we see that both transcription factors bind and form a complex with comparable affinity. So this loss of these large structures is not simply because they cannot bind as well. Now, this combined with our other data leads us to theorize that the composite motif results in these proteins to form a different conformation, which allows for them to form these solid-like condensates that cannot form when we separate the motifs and have their canonical motifs. And so with the finding that the composite motif found in these Greek island enhancers drives solid-like phase separation, we wanted to further probe the dynamics of these proteins within these condensates. Now, to study that, we performed fluorescence recovery after photobleaching, or FRAP as it's abbreviated. When we perform FRAP and we look at the LHX2 fluorescence, we see that after 10 minutes after bleaching, we do not have fluorescence recovery, indicating that these molecules are very solid and they're not moving around. We see the same effect with LDB1. When we bleach it, we see that there's no recovery after 10 minutes. Now, what's intriguing is when we FRAP these condensates and look at EBF1 signal, we actually see that the signal returns. On average, it returns to about 50% fluorescence after a couple of minutes, which is really interesting because it's telling us that these proteins have different dynamics within the condensates. And I want to remind you all, though, that even though EBF recovers, it is still much slower than a traditional liquid phase separated condensate. Typically, a liquid phase separated condensate will recover after a couple of seconds of FRAP. And so all of this data together is pointing towards the OR hub forming a very rigid, solid-like condensate. And this in vitro data was really intriguing, but we wanted to apply it to in vivo and to our system in OSNs. And so unfortunately, I don't have time to get into the details of this today, but I just want to really describe in brief the work we've been doing in OSNs. So we've been collaborating with Joan Palupa, a postdoc, and Olga Stati, a tech in the Lombardis lab. And they've done really incredible work where they've managed to culture olfactory sensory neurons and image them live over time. Furthermore, they've developed a system to label the transcription factors at endogenous levels and to label the active OR gene to image the active hub. Now, again, I don't have time to get into it today, but their live imaging confirms that these factors all co-localize together in live OSNs and that they show very slow, reduced dynamics compared to traditional liquid phase separated systems. And so all in all, we're proposing that the OR transcriptional hub is forming a solid-like phase-separated condensate mediated by these factors binding to the composite motif of enhancers. And we propose that this solid phase separation is the key to the OR hub staying stable and staying maintained throughout the lifetime of the OSN, which requires continuous OR transcription. And we propose that this model could be generalizable to other systems. As we see more and more examples of systems which rely on extremely precise long-range contacts, we think that we might see phase separation in more rigid contexts appear in other systems. So in a traditional model, we have transcription factors bouncing along their motifs within their topologically associated domain. They can start to accumulate as these motifs are found very frequently next to each other. However, in systems like the OR hub, we have motifs that are very few and far between. And so we propose that these proteins take on a very specific conformation, which allows for their solid phase separation, therefore their maintenance of very stable long-term DNA contacts. And so with that, I just want to thank everyone in the Lombardis lab and thank Stavros Lombardis, my thesis mentor. I also want to thank Joan Palupa and Olga, who have been doing a lot of work in live OSNs and really making this a beautiful story. And then I want to acknowledge the imaging cores at Columbia that have helped with all of this, and also my co-advisor for my thesis, Lawrence Shapiro. And I also just want to thank 4DN for hosting and welcome any questions with our remaining time.